Now we're going to move on to the second keynote presentation titled From Lab to Market, the Journey of Commercializing Innovations. Now let's welcome on stage Professor Dennis Lo, SBSJP, Vice Chancellor and President of the Chinese University of Hong Kong, and deliver his keynote address. Please welcome Professor Dennis Lo.
So thank you very much. It's my great honor and privilege to be speaking at this distinguished event. So I'd like to thank Professor Sunny Lee for inviting me, Mr. Ivan Lee, Mayor Liu, Mr. Peter Yan, and Ye Bujiang, distinguished guests. So today I'd like to share with you my story about how to commercialize a university-based invention and some of the challenges that we have faced. This is a picture of myself taken over 30 years ago when I was a medical student at Oxford. So as a medical student, we have to study different subjects, and one of those subjects is obstetrics. So I learned about how when a woman is pregnant, she'd like to know about the health of the baby, including the genetic health. But at that time, to sample fetal cells for DNA and chromosome analysis, we have to stick a long needle into the uterus. And every time we do that, there's a risk that we might harm or even kill the baby. So as a young medical student, I was thinking, is it possible that I can do away with all of that to develop a non-invasive blood test that I can take from the mother? So I was thinking, is it possible that a baby might release its cells in the bloodstream of a mother? But the question is, if this phenomenon exists, how do I know the cell is from a baby? So I thought, well, if the baby is a boy, then maybe we can start to see male chromosome positive cells in the baby's blood. So I persuade a professor in Oxford to actually allow me to do the experiment. As you can see from this slide here, we use a probe which, when bound to a male cell, will give it a black dot. So here you can see a number of male cells in the blood of this mother. So showing that my thinking is correct. But you can imagine if every time I have to do a test like that, I have to find those cells, it's very tedious. So thinking, is it possible I can automate that somehow? So at that time, I went to a lecture, was given by a professor called Sir John Bell. So we talked about a new method at that time called PCR, polymerase chain reaction. He talked about this method is going to change the world. So after the lecture, I actually asked him to teach me this method, which is very sensitive, which can detect DNA. So then I was able to actually use the PCR to detect those male fetal cells in mother's blood. So I published this paper in The Lancet just on the year when I qualified as a doctor. And then a few months later, The Lancet actually published an editorial called Is It a Boy? Because theoretically, you can actually use this method to tell the sex of a baby. And then I went out to work as a junior doctor for 1 year. But I thought that because I've started this field, I have some responsibility to finish it. So at the end of that 1 year, I decided to actually go back to Oxford and to do this full-time as a PhD student. So here you can see a picture of myself, actually all covered up, because I'm using male cells as a marker. And of course, every cell in my body is male and it has a Y chromosome. So I'm trying to protect my samples from myself. But sometimes in life, when you do something part-time, it's all very fun. But when you do it full-time, then you start to see all the trouble. So similarly in this story, when I start to work on this project full-time, I start to see all the problems because even though there are fetal cells in mother's blood, they are really very rare, like trying to find a needle in a haystack. So basically, after 3.5 years of work, I failed. I couldn't actually find a test which would work. But at that time, the science didn't go well, but luckily, I actually met my wife. So we actually graduated together in July in 1994. And then I went back out to be a doctor and then work until 97. So that was a year when Hong Kong came back to the sovereignty of China. And so I thought it was time to come back. And then I joined the Chinese University of Hong Kong. And then I was able to think about what have I done wrong for the previous 8 years? Why couldn't I solve this issue? Then actually about 3 months before I came back, in this journal, I saw there are 2 papers which says that in patients with cancer, you can actually find cancer DNA in the blood. But more specifically, it's not actually in the blood cells, but it's actually in the fluid part of blood, which is plasma. And then I started to think, the baby living inside mother is actually a little bit similar to a cancer growing in a patient. So maybe I've been looking at the wrong place. Maybe I should be looking at the cell-free part of blood. But the problem is that when I first came back, at that time, the funding for research in Hong Kong wasn't as good as now. So I didn't have a lot of resources. So I can only do something very simple. And I actually have never worked on plasma before. So question, how do I extract the DNA from plasma? Then I remember actually the time when I was in Oxford, I never actually enjoyed the food in the college. So sometimes I actually cook instant noodles in my room. So I was thinking, can I cook the plasma just like I cook noodles? So you take a bit of a fluid and then you boil it for 5 minutes and then take a few drops to be tested. But surprisingly, even with this very crude preparation, I was able to get a Y chromosome signal in some women. You can see the arrow there. So on the left, you can see 3 women with the signal. And then you see several without signals. And eventually, I find all the women with signals all carry baby boy. And all the ones without signals all have baby girls. So this is the first demonstration of presence of fetal DNA in the mother's plasma. And then we find that DNA is present very early, from the 6 weeks. And actually, by the 10 weeks, it reaches 15%. So I thought maybe we can use this to detect some of these disorders, like Down syndrome, which is a chromosome disorder, which is the #1 reason why pregnant women go for prenatal testing. Now, conventionally, you test for Down syndrome by taking a cell from a baby, and then you probe the cell to ask how many copies of chromosomes the cell has. So here you can see 3 copies of chromosome 21, which is abnormal. But the problem is that in my field, I don't have cells because we have DNA swimming outside cells. So how can you look at things outside cell to deduce what is happening inside cell? So it took me 10 years to think about the answer. So the answer actually is quite simple. It's like you have a wallet. Now sometimes we have 4 coins, and some other times you have 3. But I want to know how much money you have in your wallet today. So one way is to ask you to hand me your wallet. And this is like invasive testing, right? But a better way is to ask you to stand on a balance to see how heavy you are. Now theoretically, the day when you have more money, you should be heavier. But of course your balance has to be super accurate. Just like if you are 60 kilograms in weight, but the coins is only to be like 0.1 kilogram. So it has to be very precise. So what that means is I need to develop a very precise way to count DNA. Now conventionally, let's say that you have many DNA molecules in a test tube. If I count them together, the problem is that most of the DNA in the mother's blood is the mother's DNA, which is a red signal, with very little is from the baby, which is a yellow signal. So if you do that, then the mother signal is going to overwhelm the baby signal. But what you can do is to distribute the DNA into many, many test tubes so that one test tube only have 1 molecule or no molecules. So in this situation, the baby's DNA can be analyzed on its own without interference from mother. So we did that in 2007, and it works. So we very happily published that in a journal. But surprisingly, a few weeks after I published a paper, another group of people, they published the same thing. And actually, both papers were submitted on the same day, which is amazing. So that means that we have competition. But on the other hand, the good news is that if 2 people have thought about the same thing, it probably works. But however, this method still has some bottleneck. And what's the bottleneck is that when you do PCR for testing, you have to select a gene to be tested. But on chromosome 21, there are 150 genes. So by using this simple method, I cannot maximize the information. So I need to have a way in which I can analyze everything. So at that time, a new method was invented, which is a DNA sequencing approach. So I've thought about how about if I do it randomly and do it quantitatively. I just randomly sequence molecules in the mother's blood and then map it to the chromosomes and then can calculate the ratio of different chromosomes. Now, interesting, while using this method, I was able to get a very big signal in babies with Down syndrome, which is the green and the red signal. But in normal baby, it's normal. So the accuracy was very high. It's 100% in this first test. So we published that in this journal. But also surprisingly, within a few weeks of this paper, the other people, they published their work as well. So then I was very surprised. So who is this guy? So I looked at his website and find that he's a professor in Stanford. And surprisingly, he also actually studied in Oxford and actually graduated in the same year. So I went back home and asked my wife whether she knew this person. She said she didn't. But then we get back this picture. So it's me and my wife, and that's Steve Quick. So it's almost like today you're listening to my talk, and actually the guy next to you is competing with you in 30 years from now. So you better take a selfie with him or her. So anyway, and the story didn't stop there. So about 3 years later, I was elected into U.S. National Academy of Science. So when I read the report, of course I'm very happy I saw my name. But a little bit down the line, I saw him. So this is a picture that we took together at that ceremony. But anyway, so you can see now that we have a prototype of method which seems to work. But to push it out, I need to do clinical trial. So we spent the next 3 years doing, and it was 99% accurate. So we thought it's ready. But of course, I can't do it in a university. I have to work with some companies. But when you're trying to work with a company, they ask you, do you have a patent? So basically, for the last 20-odd years, we've been very diligently filing patents. Now, for example, every year, Nature Biotechnology will rank the top 20 groups in the whole world in the numbers of biotechnology patents. And for the last few years, I'm happy to report that I was on the list. Now, for example, in the latest list, I was number 7. And of course, you wonder, who are the other guys? Now, on the number 1 is Jiang Feng from MIT, who actually is one of the inventors of genome editing. Number 2 is the CEO of BioNTech. And number 3 is a very prolific inventor. He has founded many companies, including Moderna, which is another mRNA vaccine company. And number 5 is Jennifer Doudna, who also invented genome editing and got a Nobel Prize for that. And we also have another colleague, is Alan Chen, is number 13. You can see that in this list, most of the prolific inventors are from the US, with 3 from China and 1 from Europe. So I think we have to work very hard to actually push this forward. But that's now. But 15 years ago, I wasn't as experienced with patents. So my first patent, my university actually licensed it to a company in the UK. But the company didn't realize the significance of that. So they sat on it for a little while and gave it back to the university. They thought it was rubbish, which of course is a big mistake on their part because this patent turned out to be very valuable later on. So then I have to go and find somebody else who is interested. So in one of the conference, I met this gentleman who is Charles Cantor, who is the Chief Scientific Officer of Sequenom. So we talk and we collaborate, and eventually my technology was licensed to Sequenom. But in Stanford, of course, they have a lot of experience with commercialization. So they licensed technology to Verinata, which was then acquired by Illumina for $600 million. And then, of course, when patent was owned by a company, they start to fight battle. So we have a series of 4 lawsuits between the 2 companies. And eventually, I won 3 of them, and they won 1. So we settled, and they pay us $50 million US, and then we pool the patents. So now this technology is called non-invasive prenatal testing, or NIPT for short. It's been now used over 100 million times. Now, so I wonder, apart from prenatal testing, can I use this technology elsewhere? So I'm thinking, how about cancer? Because of the similarity of cancer and baby. But then cancer is a bit more difficult. So initially, we want to just look for mutation of cancer. But those mutations are usually very few and far in between, like 1 in a million base pair. So eventually, we decided to look at something called epigenetics, which is basically modification of genome. It's like you format your Word article differently. Like different fonts, different color, different size. And then we invented a method in 2013 to use this epigenetic method to tell if somebody has cancer. And also, because different organs in our body have different epigenetic profile, I can also tell you where is that